Non-Enzymatic Chemistry Journal and won various recognition awards and her Best Poster Award along with her then postgraduate student at the Niraja Bapat at the Origin 2014, the second joint international conference of the International Astrobiology Society and Bioastronomy held in Nara, Japan. She has written several journal communications in national and international review, which is worth mentioning, and is presently she is an associate professor in the IISER Pune School Lab as mentioned by Maharaj. So wasting no more time, I now request today's eminent speaker, Professor Dr. SUDHA RAJAMANI, to please start her today's lecture on the topic, the story of how life could have originated on Earth. Over to you, madam.
Thank you, Mr. Samir. I'm going to turn my video on, and I hope that's okay. Yeah, can you people see me?
Perfectly.
And can you also see my slide in the full mode?
Yes, it is visible, madam.
Okay. First of all, thank you very much, Swami Kamalasananda ji, for your invitation. I'm glad I answered the phone call, even though I did not recognize the number, because I generally don't answer phone calls of numbers that I'm not very familiar with. But I'm glad I did answer that, and I'm glad all of that led to this evening. And I'm very glad to join you, your colleagues, and your students online. I surely look forward to meeting you all in person at some point of time. I think both of your introductions of me was a little too generous. So I just would like to remind students that, you know, journey in science can be very fun and exciting, and can lead you down paths which you never even imagined when you started out in this sort of larger realm in your undergrad. So be very open to whatever comes your way, and yeah, be excited and be curious. I think that is what is going to eventually make the journey real fun. So without further ado, let me get to the main discussion at hand today. We're going to try and understand, at least from an astrobiology perspective, how is it do we think that the transition from non-life to life occurred on the early Earth? And this journey wouldn't be possible without my lab and the fun people that work along with me. This is a whole bunch of grad students and a postdoc whose pictures are here, but also there are a lot of undergrads who have contributed to the work that happens in the lab. And we basically are very interested in questions that exist at the edge of geochemistry, prebiotic chemistry, which is the chemistry that occurred before life is thought to have emerged, and minimal life. So in this particular realm, we ask the question, how molecules in a prebiotic soup, which were plausible because of certain chemical processes and geological processes, how do you go from those molecules in the monomeric space to form polymers, which essentially is what extant biology is made of? All your biopolymers are polymeric in nature. They are monomers strung together to form either proteins or nucleic acids or carbohydrates, or in the case of lipids, they are not strung together, but self-assembly processes driven by thermodynamical constraints essentially force them to come together to then evolve properties or sort of what we call, they have emergent properties which cannot be ascribed to the monomers themselves. So whenever the monomers are in isolation, they do certain things, but they come together and form these polymers, and they start doing things which now push them to the realm from chemistry to biology. And one of the important things in the lab is to try and understand how this transition happened. And as we go, it'll sort of become clear how we think about this. So the origin of biomolecules, if you kind of look at why we think about this transition from chemistry to biology, it will become very apparent in the slide. Did the slide change? Does it say origin of biomolecules? Can you see it?
Yes, ma'am, it
Yes.
is visible.
Yes, ma'am.
Oh, okay, great. So the slideshow's working.
Madam, I'm interrupting you, please. Please hide the small pop-up window at the bottom.
I'm not sure how much I can hide. If I hide, I'm worried everything will go away. Let me see. See, this is what happens.
Oh, yes.
You know what I mean? There is some kind of a weird thing that happens. Can you see this now?
Yes, it's visible.
Okay, wait. Let me try this
Yes,
again. Can you see this now? Perfect.
Yes, it is visible now.
Awesome. Great. So
Thank you.
when you look at the slide, you start appreciating this beautiful continuum that exists from physics to chemistry to biology. So in the bottom panel, if you can see my arrow, right? You let me see if I can do a laser pointer. Yes. Can you see my laser pointer? Yeah.
Yes,
So
madam.
awesome. So this bottom panel is basically highlighting some of the molecules that you see in biology today. You have a bunch of amino acids here. You have a protein here. You have an RNA enzyme here called the hammerhead ribozyme. Fundamentally, if you zoom into these molecules, what are they? They are essentially large ensembles of chemical entities. Yeah. And for example, your proteins are made up of monomeric entities called amino acids. Your nucleic acids or RNA and DNA are made up of monomers called nucleotides. Now you zoom further into them, what are they? They are essentially a conglomeration of atoms which are held together by transient and non-transient bonds, essentially chemical molecules. And this chemistry is not restricted just to life on Earth. If you actually use these very high-end spectroscopic instruments and look at the space, in the interstellar medium, people have detected molecules which are very relevant to what we see as life molecules on Earth. For example, people have looked at meteorites and they have found that you have a whole inventory of amino acids present in them, many of which are not represented in life today. What this means is the chemistry that is relevant to life is happening all over this universe. So what makes Earth so special that we harbor life as we understand it? And I'm going to highlight this, as we understand it, right? And not just simple molecules. There have been also large complex molecules called buckyballs, which are essentially C60 carbon atoms containing molecules that have been discovered in space. Now zoom into that, and what are they? They are essentially made up of a bunch of atoms. So if you can appreciate this continuum from chemistry to biology, it will become apparent to you why is it that we try and understand this transition from non-life to life as a transition from chemistry to biology, which is what we do in our lab, and we do it in a certain specific context. So where do we think these biomolecules might have come about from? So there are two hypotheses in the field. One says that a predominant amount of molecules that you see in the prebiotic soup, the soup that was present very early on Earth, where interesting chemistry was happening that led to the emergence of life, a lot of the molecules in those pools were essentially molecules that rode on comets and meteorites and were transferred onto Earth when there was a lot of bombardment that happened. Even today, you have meteorites dropping on Earth, and if you actually take some of these meteorites, and there's this meteorite called the Murchison Meteorite, which fell in Australia in the 60s. My mentor from UC Santa Cruz, Professor Deemer, actually worked with that meteorite, and they went on to show that the core of the meteorite do have very interesting organics like hydrocarbons, like amino acids. So even to date, this delivery happens, but back around 3.8 billion years ago, there's supposed to have been a period called the late heavy bombardment period, where Earth was constantly being bombarded. Earth and Moon were constantly being bombarded with meteoritic delivery. And it's thought that a lot of these interesting molecules that can form in the interstellar medium were delivered by that process. Also, terrestrial environments like the ones that are shown here, these are terrestrial geothermal pools, which exist at the edge of volcanoes, which are rife in a lot of chemical and thermal fluxes, which allow for interesting chemistry to be plausible. So if you have a set of chemical ingredients, simple molecules, they can come together and form more complex molecules like the ones that are listed here: formic acid, urea, serine, glycine, some of your amino acids. So it's thought that by these two processes, biomolecules could have been delivered on the early Earth. And unfortunately, there's probably not too much interaction possibility, but I would like an answer from at least one of you. You see a bunch of images here, preferably a student. Sorry, I would like a student to answer this question, or students to answer this question. You're seeing a bunch of images here. You see T-Rex, you see mushrooms, you see these single-celled bacteria, then you see a very giant catfish, and you see some of the tallest trees in the world. These are called redwoods. And then you see a human giant, Nelson Mandela. What do you think is common to all of these entities whose picture is present on the slide? Anybody?
Ma'am, it's Africa. Ma'am, it's Africa.
These are all in Africa.
Did you say Africa?
Yes, ma'am.
No. There is something much more common to them, which has nothing to do with geographical location. It's got to do with biology.
Ma'am, DNA.
Cells. Heard that answer.
Ma'am, DNA.
Made up of biomolecules.
Cells. Biomolecules, fundamentally, they're all made up of cells. Yeah. Cells are made up of biomolecules. So this is the least common denominator between all of these entities that are present in these various pictures. So one of the interesting ways, or simplest ways, and most profound ways of asking this question of how the transition happened is to try and understand how the fundamental unit of life came about. And the fundamental unit of life is not a simple unit, right? I'm showing two images here, a prokaryotic cell and a eukaryotic cell, and they are super complex. Even the prokaryotic cell, which is seemingly super simple in comparison to eukaryotes like us, is still very, very complex. It's made up of a whole bunch of things, as mentioned here, right? So what would be the best way to articulate this question of transition of non-life to life? It's to ask, how did these cells come about on the early Earth, given all the interesting chemistry that was happening? And why is it a very relevant way to pose this question? Look at these cellular entities even today. You see them populating every possible niche on the Earth. UV resistant organisms. They can take kind of UV radiations that you and I will completely get mutated and we might not even look like us. Rather we'll all be gone, right? There are extreme heat-loving organisms, thermophiles. There are salt-loving organisms that can tolerate super high content of salts, where you and I will have our cells shrivel and die. These are called halophiles, permafrost-dwelling organisms. These unicellular entities have literally populated, habitated every plausible niche on the planet, begging the question, what makes them so amazing? How did they go from being a bunch of interacting molecules to entities that went on to populate every single niche that you can think of on the Earth? So this is the quintessential tree of life I'm showing an extremely simplified version of it, which essentially probably says that there was a last universal common ancestor from which you had the three domains of life come about: the bacterial domain, the archaeal domain, and a crosstalk between these two domains eventually led to the formation of eukaryotes, from where we finally evolved. This is a very sort of a minimalistic version of the tree of life. However, this thinking is increasingly being questioned, and what is coming up, what is sort of emerging from this, from the various things we have learned in the last couple of decades, is that possibly this is how the tree of life looks. It's much more complicated, and way at the base of the tree of life, it's not like this one single last universal common ancestor, but it probably was a common ancestral community of primitive cells or protocells, which were lifelike, had all the hallmarks of life, and some selection pressures at some point created this bottleneck, which then eventually caused the emergence of the three domains of life. Yeah. Now, what are these protocells that I'm talking to you about? They are fundamentally just like our modern cells, but much, much more simpler. They also are comprised of lipid membranes, but the lipid membrane structure here is like a tennis ball. And if you cut open it, what you see is the inside of the tennis ball. The outside is made up of this bilayer lipid membrane, and the inside has essentially a genetic material that is encapsulated. Now, the genetic information in your cells and my cells are encoded in deoxyribonucleic acid. But that is not the only genetic material that is possible, right? Because you have viruses, just like our the SARS-CoV-2 virus, which has completely upended our lives and turned it topsy-turvy, is a RNA virus. It is a virus in which the genetic material is encoded in an RNA, which is a cousin of DNA. So these protocells are thought to be these lipid bags which encapsulate some version of a genetic material and some minimal metabolic network that can essentially use the sources from the environment to make energy and to keep themselves far away from equilibrium, because life is a non-equilibrium process, which is far away from equilibrium. And this is sort of an artistic rendition of the protocell. And the idea is to give you a sense of the bilayer lipid membrane that surrounds the cell. And inside, it's got a whole bunch of biomolecules, some which encode information and some which are the energy-making entities which form networks and use reactant substrates to form products that essentially allow the cell to exist, to survive. And this is a very interesting sort of a sentence from an article that some of my colleagues put together, where they encapsulate what is it that we understand about this whole transition from non-life to life. The best we can ever do is to draw up the story that is consistent with data that we have, with actual evidence, right? Where do we get the evidence from? Experience in geology, which tells us about how the early Earth environment was, some experimental and some theoretical, with what chemistry reveals about the molecules of life, and with what biology reveals about some of the oldest forms of life, which I showed basically have taken over every niche that you can think about. And happy to say that after more than a century of effort, this story of how this puzzle works is coming into view. Do we have all the pieces of the puzzle? The answer is no. But is it something that we can sort of logically think about what it might be? The answer is yes. And as Maharajji already mentioned, it's an extremely interdisciplinary area, and astrobiology, which essentially helps us understand how life came about on our planet and elsewhere in the universe, and what its distribution is probably like elsewhere in the universe. It feeds off of a lot of different areas of sciences: chemistry, biology, geology, paleontology, ecology, astronomy, physics, fundamentally all the way from cosmology to biology, and feeds off of all the sciences in between: physics, chemistry, and math is also, math underlies all of physics and all, right? So it's a very, very interdisciplinary field. And in our lab, we are interested in understanding how do you go from the prebiotic soup of molecules to these protocells, which eventually evolved into more complex cellular entities, finally leading to life as we see it. And what are the scientific areas whose help we take to answer these questions? Biochemistry, molecular biology, biophysics. We work with colleagues who do experiments on the computers. They model reaction systems, right? We use their expertise. We work in the realm of organic chemistry, or more specifically prebiotic organic chemistry, a lot of inorganic chemistry, geochemistry. All of these areas of research, tools from all of these areas of research fundamentally help us understand these processes that we're interested in, predominantly this thing here. How do you go from this space to this protocellular space? And here is a slide which gives you a sense of the early history of life on Earth. It is thought that about 4 to 4.2 billion years ago, a lot of interesting chemistry relevant to modern biochemistry was already happening, leading to the fundamental monomeric entities that were required to allow for this transition to happen. From that space, about 3.8 billion years ago, it's thought that the RNA world came about. And why is this RNA world very interesting? It kind of simplifies our life a little bit. In modern biochemistry, you see that the information is encoded by DNA or RNA, and RNA processes all of this information and makes proteins, which in turn make more of DNA and RNA. It's like the cyclic loop. It's like this chicken and egg paradox. Which came first, right? Did the DNA come first or the protein come first? So now if you think about an RNA world space where this cool molecule can not just encode information, it can also catalyze reactions like proteinaceous enzymes. These RNA enzymes are called ribozymes. You now have a scenario where the problem is simplified, and then you're trying to see how do you go from this prebiotic chemistry space to an RNA world space. And work in the last 15, 20 years has made it very apparent, including work from our lab, that going from the prebiotic chemistry space to an RNA world space is non-trivial, and this might have been predated by pre-RNA worlds where the molecules look a lot like in their sort of overall structure and function like probably the RNA world molecules, but they're much more easier to make, because making the RNA molecule directly from the molecules in the prebiotic chemistry space from the prebiotic soup is completely non-trivial. So what are the kinds of environments that are thought to have supported this transition, right? There are again two schools of thought: the hydrothermal vents, or the black smokers, which are present, which are essentially geological activity, volcanic activity on the ocean floors, also harbor this very interesting temperature and chemical fluxes. It's thought that these environments might have allowed for the transition. In our lab, we kind of belong to the school of thought because it's a little more tangible for me. We think that terrestrial geothermal pools, like the ones which exist at the edge of volcanoes, where very interesting chemical fluxes are happening, temperature fluxes are happening, pH gradients are present, can allow for a lot of these interesting oligomerization reactions that I was talking to you about. How do you go from amino acids to peptides and proteins? How do you go from nucleotides to nucleic acids? Some of these reactions we think would have been readily been favored here because of very nice geological processes that can be sort of supported in these scenarios. This is just images of two of these things from, even the previous one had an image from Mount Lassen National Park, but this is just to show a hot spring from Kamchatka, and here is Yellowstone National Park, a hot spring. We have some of these hot springs in India too. Unfortunately, they are not maintained as well as these sites in other parts of the world, but we are still working with some of these analog site samples that come from Ladakh, and I will touch upon it towards the end. And what is the model that we work with, right? I'm talking to you about all these things and the transition. There should be a working model that we should work with. What is that model that we think that might have, that might explain the terrestrial origins of life? It's thought that if you had can you see the slide, the model for terrestrial origins of life?
Yes, ma'am.
Awesome. So if you have the fundamental monomeric entities that can form your informational polymer and your bag of lipid bag or the cells layers, multiple wet-dry cycles can facilitate some of these reactions that can allow for the emergence of protocells. Why are wet-dry cycles even plausible? Just like on modern Earth, wet-dry cycles would have been driven on early Earth by day-night cycles, seasonal cycles, yeah, and by tidal activity. For example, when you have high tide at the edge of these water bodies, you can have lot of water activity, aqueous activity. When you have a low tide, water recedes and you can have a dehydrated scenario. So these kinds of geological processes would have been very readily sort of facilitated even on the early Earth. And these wet-dry cycles in these realms, in these niches, are thought to have allowed for the transition from the monomeric space to the polymeric space. So for example, during a dehydration phase, if you had these lipid molecules, monomers of the lipid molecules and monomers of the RNA molecule present, these lipid molecules, which can very readily assemble into higher order structures like micelles and vesicles, you put them in water, they're water-hating groups. They are made up of water-loving groups and water-hating groups, right? These fluorescent blue-green are the water-loving groups. The black rods are the water-hating groups. Here are structures of two single acyl fatty acid and fatty alcohol systems. Take a bunch of these molecules and put them in water. The water-hating groups will immediately push the water out and ball up into these balls, either a micelle or a vesicle. And when you dry them down in the presence of other solutes, they readily form these multilamellar sandwiches. And in each alternate layer where you have the hydrophilic head group, if your solutes are polar in nature, they can actually get trapped in those spaces. So now, instead of thinking about forming these bonds between two monomers, which requires the removal of water, which is not going to happen in a very water-ridden place, right? You need some kind of dehydration event to pull out water. So now when you have this dehydration event, you not only pull out water and reduce water activity, you also constrain the molecules from a three-dimensional space to a two-dimensional space, where the interaction between molecules becomes much greater. And with multiple cycles, you can have these long strings of red things that you see here. These are essentially supposed to be the informational polymer. They can form readily, and during a subsequent rehydration phase, these lipid molecules again bleb out into these cell-like entities, encapsulating these growing polymers. This inset here actually shows you a real microscopy image of lipids that we can work with in the lab. These are single-chain fatty acids called decanoic acid, and when you mix them with a fatty alcohol, in this particular case a C10 fatty alcohol, they very readily assemble into these vesicles, which as soon as you hydrate them, they start blebbing out into this lovely tubes and bulbous structures, and they can encapsulate the solute that you have in the buffer. And in this case, it's a fluorescent molecule. So it's thought that multiple such cycles are thought to have led to the emergence of protocells, which can now undergo even a primitive form of selection over multiple cycles, because some of the encapsulated oligomers can maybe bind to the lipid vesicles and kind of make them more strong and prevent them from breaking apart, like this one here. So it's thought that these kinds of cycles eventually would have led to those ancestral community of cells that I was talking to you about, that is present at the base of the tree of life. When did all of this start, right? How did this field even come about? It came about because of thinking of philosophers that dates back to several hundreds of years. I'm not going to talk about them because the scientific paradigm was essentially put in place by these two very famous scientists. And one of them has a West Bengal connection. Yeah. Anybody recognize this gentleman? This is JBS Haldane. So Haldane had a very interesting West Bengal connection. This is Oparin, who was a Russian scientist, a Russian physicist. Both of them, back then, no internet, no email, nothing, right? Independently, the two of these gentlemen in Russia and in America came up with this very interesting hypothesis where they said that given the right kind of ingredients, like methylene here, dicarbon here, water here, ammonia, carbon dioxide, given the right kind of chemical ingredients in this vast ocean, prebiotic ocean that was present, if you had a source of energy, either in the form of lightning or ultraviolet radiation from the sun, they can facilitate some very interesting chemical reactions that would have allowed for the transition from this chemical space to a more biological space. In this particular case, they are using an example of formation of polypeptides, which are made up of amino acids, which emerge from the reactions that are possible between these chemical entities. So particularly Haldane proposed the term prebiotic soup or the primordial soup, which has led to cartoons even several decades ago. But the idea is that if you have the correct ingredients in the prebiotic ocean, in the vast prebiotic ocean, it provided a fantastic laboratory for interesting chemistry to happen, which allowed for the transition from chemistry to biology. But a hypothesis is a hypothesis till proved with an experiment, right? Systematically, till you prove this whole hypothesis with an experiment, it still is a hypothesis. So one of the first proof of principles came from the Miller-Urey experiment. This coincidentally happened in the same year as your structure of DNA. The DNA structure was elucidated in 1953. In the same year, Miller and Urey actually went on to show that if you have a bunch of inorganic molecules like methane, ammonia, and dihydrogen in the presence of water, here is the simulation of the prebiotic ocean in this little setup that they put together. They subjected it to heat, and the gases vaporized, and the chemicals went along with the water vapor into the space where they simulated lightning using an electrode, tungsten electrode. And they went on to show that interesting reactions in the space essentially led to the formation of some of these molecules that are shown here. There's a condenser here, and that condenser can again condense the gases into liquid, and you can collect the liquid for analysis after multiple cycles, right? They kept this experiment going for, actually, if I'm not wrong, several months and even a few years. Stanley Miller was extremely rigorous about keeping notes, saving samples, to the point where his first graduate student, Jeff Bada, and one of his second last graduate students, who's a good friend of mine, Henderson Cleaves, they got together, they took out the samples from 1953, later on, early 60s, late 60s. Stanley's group did a lot of different kinds of versions of these simulations of this particular hypothetical situation. And they once again reanalyzed these instruments, these samples with very high-end instruments, and went on to show that you not only form simple hydroxy acids and the simpler amino acids, you even form in parts per billion some of the more complicated amino acids like phenylalanine, tryptophan, tyrosine, so on. Yeah. So very long story cut short, starting from 1953 till about 2015, 2016, a lot of interesting studies have shown that the same starting ingredients can lead to the precursors that eventually lead to the formation of amino acids and proteins, nucleosides and nucleic acids, and lipid precursors that leads to the formation of protocellular membranes. This is a paper, very cool paper, if you guys can lay your hands on it. It is by the Sutherland group. They showed this sinosulfuric metabolism that allows for the formation of precursors that leads to 3 different biomolecule classes. So suffice to say, the prebiotic soup would have readily had all of these molecules present. So how did you then go from those molecules to protocells is a question that is still unanswered. But subsequent to that, it is thought that these ancestral community of cells, which were capable of different processes of life like growth, division, and undergoing Darwinian evolution, eventually led to your earliest prokaryotes, cyanobacteria, which basically did one very important thing on the planet. Prior to their emergence, the planet was this really, really hot, crazy, and intensely sort of, it was ridden by chemical parameters that won't make us believe that life could have been possible, but the common ancestral community of cells is thought to have still come about in specialized niches that did allow for these transitions to happen. And many of these reactions that we try and do in our lab are from 60 to 90 degrees C, so they're very readily plausible. But eventually, they're thought to have led to the formation, emergence of prokaryotes, which did something very, very important that completely changed the way subsequent life could evolve. What did they do? They oxygenated the earth. Prior to their emergence, the oxygen level was really, really low. Even now, if you think about it, what is the major gas in the atmosphere?
Nitrogen.
Nitrogen.
Right? What percentage of our atmospheric gases is oxygen?
78%. 78%.
Ma'am, oxygen 21%.
Exactly. 78 is nitrogen, right? Now 70 something is nitrogen, 21, 22% is oxygen. Where did all of this oxygen come from? You will have to take my word for it. There is enough geological evidence to show that all of the oxygenation that you see on the earth now was driven over billions of years by what look like pillows of rocks, right? These are actually cyanobacterial mats which have interacted with rock and minerals and form these pillow-like structures called stromatolites. This is present even to date. It's a modern image. It's not an artistic rendition, right? It's an image from Perth, Australia, Western Australia, where you have them present. You take them and you cut open them, and you will see these alternate layers of geology and biology evidence still very much existing. And these cyanobacteria-like entities are thought to have essentially oxygenated earth that we see. And when did this happen? We saw that the transition from prebiotic soup to maybe the emergence of RNA world was between 3.8 and 4.2 billion years ago, right? So the oldest geological evidence of life here, it says about 3.5. Now we can say that it's actually 3.85 billion years ago, because some of my collaborators who work in University of New South Wales and who work in, they are geologists and who work with some of these systems of rocks and some hot spring deposits have actually gone on to show some of the oldest geological evidence of life, and they have traced it back to 3.85 billion years ago. So if you put a clock, yeah, if you take that origins of life ka jo linear bar maine dikhaya and you make it into a 24-hour clock, with 3 o'clock being morning, 6 being noon, 9 being afternoon, and 12 being midnight. Midnight is when your earth came about and interesting prebiotic chemistry and all happened. Some of the oldest definite fossils of prokaryotes emerged around, the signs of it are from this space. So around 3 to 4 billion years ago is when all of this was happening. And what did they do? They oxygenated the Earth's atmosphere. They took ingredients like carbon dioxide and water from the environment. Metabolism happened, and they fueled their existence and spit out oxygen, which is what, of course, all of this happened with light energy. And this is what is thought to have led to the increase of oxygen in the atmosphere. Subsequent to that, about 2 billion years ago, so all of the saga was going on around 3.85 billion years ago, right? About 2 billion years ago is when the first eukaryotes are thought to have emerged and the first multicellular life subsequent to it. So we are already now looking at close to afternoon, right? Midnight ho gaya, morning ho gaya, noon ho gaya, we are in late afternoon now. This is when even our ancestors came about, the eukaryotes. This was, like I say, about around 2 billion years ago, where they had membrane organelles, as against the prokaryotes, where all the molecules were predominantly interacting in sort of one bag of cell, even though a lot of modern research is showing that there is some kind of gradation of the concentration of these molecules across the cell, and there are still these phase-separated areas where things would have been present. In your eukaryotes, you actually have a whole bunch of elaborate membrane-based organelles where things are more contained and several things are happening in these smaller regions, and then there is a crosstalk between all of them, and the grander biochemistry happens in the larger cell, right? All of this 2 billion years ago. And here is a choanoflagellate colony. They form these rosettes where they are held by a jelly-like structures, and these are very interesting because they are, to date, they are kind of being studied extensively to understand how our ancestors, the first multicellular entities, came about. So what do we think are the broad steps that led to the emergence of the first protocells? Interesting prebiotic chemistry leading to the formation of building blocks, like I showed you. It's then thought that a lot of the niches that I talked to you about, they would have been present in various parts of the earth. They would have led to very interesting, they would have supported a lot of interesting geological wet-dry cycles that led to the non-enzymatic polymerization of these oligomers. I specifically say non-enzymatic because as soon as you say protein enzymes, then there's the chicken and egg problem, right? Which came first? So it's thought that in the absence of enzymes, also you can actually have these monomers oligomerized readily to form polymers. And once a polymer of a certain length came about, which could encode some function, some kind of catalysis, you basically would have had a replication kick in where information in one molecule, one string has to be sort of, if there are a whole bunch of oligonucleic acids, and then some of these evolve functions, unless and until you can propagate that information, that particular thing can be lost. So non-enzymatic replication is thought to have led to eventually a space which led to the formation of the RNA world, where many more complex functional ribozymes came about, and their crosstalk with primitive membranes is eventually thought to have led to the emergence of protocells capable of Darwinian evolution. What is it that we do in our lab? We're very interested in understanding the origins of the informational molecules and also understanding how protocellular entities self-assembled, what are their physicochemical properties, what did the amphiphilic landscape look like? Your modern cells are the modern cells which we are made up of membranes where you have proteins interacting with membranes, and the fundamental unit of these lipid membranes are also very complicated molecules that are formed at the end of a very orchestrated metabolic process. And when we are talking about protocells, we are talking about geological processes that led to the formation of much more simpler amphiphiles. So we are very interested in understanding what was this landscape of amphiphiles like, and what kind of amphiphiles would have had a selective advantage to have survived those harsh conditions, and what were these protocellular entities physicochemical parameters like, how did they survive under those conditions. These are a lot of things that we think about quite a bit. We also try and understand the non-enzymatic information transfer process. We like to understand their rates. We like to understand what are the fidelity of these processes. If you look at information transfer in cells today, where there is a host of machinery and a huge orchestration of this particular process, your fidelity is like super high. It is like 0.99 something. Yeah, because errors lead to mutations, mutations lead to diseases. So you cannot, the idea is to try and minimize that to the maximum possible extent. And now when you go back to the origins of life on Earth, you don't even have machinery which can do all this complicated stuff. So then the question is, how do we think about these non-enzymatic processes? What might have been the chemical molecules which were relatives of our modern nucleotides, but were slightly different and were intrinsically active to undergo some of these processes much more readily, and were they sturdy enough, could they survive the harsh conditions of the early Earth? We are also interested in understanding how the first enzymes came about, minimal enzymes came about. If you look at your modern enzymes, a lot of the catalysis or the structural integrity of the enzyme you can attribute it to the metal that's associated with the enzyme. So think of metals. Where did metals come about on the early Earth? You have a whole bunch of minerals. Mineral catalysis is thought to have been very early version of, enzyme is thought to have been driven by mineral catalysis and their close interaction with some of the earliest molecules. We are interested in trying to understand what that sort of process would have looked like, and what are the molecules what are the kind of molecules that would have allowed for that to happen. And finally, all of that we do, we also do one other thing. We just don't sort of work in laboratory simulated scenarios. That is the huge chunk of the work, right? That is about 75% of our work, where we simulate these early Earth conditions in the lab and try to understand how these transitions would have happened. But one other thing that we are increasingly challenging ourselves with is to try and understand how would all these reactions pan out if you now start working in a much more realistic scenario. If I told you that we initially understand origin of informational molecules, where we are looking at just the monomers, and then we simulate the earlier conditions like that would have been present in a terrestrial pool, and we do wet-dry cycles, and we see how these molecules go from nucleotides to nucleic acids, and completely ignoring the complexity of the prebiotic soup. Now, prebiotic soup would have been crazily complex, but and working with that crazy complexity is difficult. One, because it just makes everything hodgepodge and you cannot tease out the fundamental processes. It becomes too complicated. Another important thing is we still do not understand the actual complexity associated with the prebiotic pool. So what we are trying to do is to use analog environment samples from hot springs and stuff like that, and increase the complexity of our starting reaction mixtures, because now you're going to have anions and cations that leached out from the rocks that interact with the water in these hot spring samples. They have very interesting effects on these processes, which you would not at all account for if you're working with very, very strict laboratory simulated conditions. And these kinds of reactions, understanding these reactions, are very important in the context of habitability. This has become a very sort of cool word these days, because a lot of you must have heard about Perseverance rover, right? Perseverance rover has a whole bunch of interesting instrumentation which is being used to understand what kind of molecules are present on Mars, what are the kind of conditions that might have been. Mars is thought to have at one point hosted life, which got completely wiped out. So the question is, what kind of signatures has that left? So habitability essentially is an area of research which is very actively being pursued, both at the level of trying to understand what are the exoplanets that are out there that have Earth-like conditions, and if any of them might even harbor anything remotely close to life as we understand it, or life as we still don't understand. I mean, that's very complicated. So these are the various aspects that we work on in the lab. And here is this Mars 2020 Perseverance rover that I was talking to you about. It has a whole bunch of gizmos, right? You have this mast cam. It allows you to study the mineralogy of the surface. You have the super cam, which is now going to actually look at the chemical composition of the minerals. And you have this, what they call pixel, which is this fluorescent spectrophotometer. And they again are going to allow for very nuanced elemental analysis of different samples that the Perseverance rover can pick up. Then you have what is called Sherlock. I think this is one of my favorite acronyms among the lot. It's basically a spectrophotometer, uses an ultraviolet laser. And again, all of these are trying to look for signatures of organic molecules that are relevant to life as we understand it. And this is something that has gotten, I think, a lot of people across the world excited about astrobiology. People who didn't know before about what astrobiology as a field was have now become cued into the word and are thinking about these things. And yeah, the hot springs that I'm talking to you about, these exist in Ladakh. They don't look as sexy as the ones I showed you, right? The Kamchatka hot spring, the Yellowstone hot spring, the Mount Larsen hot spring is spewing out water, and it's like super, lot of action happening. Here, our Indian hot springs look a little blah, but they are nevertheless very, very important analog sites. These are from different regions of Ladakh. And we did this expedition in 2016 where we went with a group of scientists who came from the US, from Europe, from Australia. And it was sort of a collaborative effort between Mars Society Australia, NASA, and Birbal Sahni Institute of Paleosciences and IISER Biology, IISER Pune, but essentially our lab. And we went out for the first time. We donned hats of being experimental field experimentalists, right? So we went out, we learned how to sample from these sites, and we tried to avoid all the sites which were too colorful. Too much color means life. Life is very colorful. Too much color, life. So we went to this really blah looking sites where the origin of the pool was basically, and this is basically thought to be driven predominantly by geochemical processes. And we got back samples from there, and we tried to look at some of these interesting reactions that we carry out in the lab in more realistic conditions, and we're getting a lot of cool answers. This is slightly older picture. This is my first student, my second student. They kind of almost joined back to back. They both have graduated and gone on to do very cool things. He's very interested in science education and science outreach, likes to think about how science has to be taught at middle school and high school levels. So that is what he's doing now. Nirja is still in the academia and she's pursuing research. These are currently my three most senior grad students who are working on multiple aspects. He loves lipids. He loves to work on the crosstalk between lipids and amino acids. She likes to think about ancient enzymes and also think about some of the informational polymer things. He's the only organic chemist in the lab. He's a postdoc. He helps us with synthesizing a lot of these molecules which are not necessarily relevant to life today, but are very important to understand pre-RNA world kind of a space, right? And some of the undergrads who have rotated through our labs. Again, an old picture. I have 4 other new grad students since then. But I thank you for your time. And I want to leave you with this thought that the origins problem can be extremely naughty, right? But it's not insoluble or insolvable. It's a matter of time, and we will probably be able to fill in the remaining pieces of the puzzle. Thank you for your time.
Thank you, madam, for your splendid, thought-provoking, and enthusiastic lecture. And you have beautifully explained the different life-forming entities and the possibilities of different chemical reactions that promoted the existence of life and its diversification of Earth. It was a really interesting journey that you have explained and about your research labs that is presently doing researches in different areas of astrobiology. Thank you, madam, once again for your beautiful lecture. Now it is the time for the audience to post their questions, and you are free now to post your questions directly to madam. So if anybody feel that there is any difficulty in understanding or you have any question in your mind, you are free to ask madam directly. Okay. My dear students, it is open to all of you. Ask one by one.
And some of the most fundamental questions are some of the most seemingly simplistic questions, but they don't have direct answers. So yes, you should ask your questions.
Good evening, ma'am.
Hi.
Good evening. My name is Ankit Bhattacharya. Ma'am, I just want to know, ma'am, that when the life originated on Earth, it was mainly mainly non-oxygenic species developed first. Ma'am, why did they change to oxygenic species? Did any mutation happened or something other?
So the photosynthesis thing that I talked to you about, when the first cyanobacteria came about and they were in a space where they literally evolved a metabolism that was allowing them to use the water and carbon dioxide from the atmosphere, and in the presence of light as a fuel, they were making these important molecules that is required for their sustenance, and oxygen literally was a byproduct, right? And this byproduct, suddenly the levels of this byproduct started growing bigger and bigger in the atmosphere. What it ended up doing is it created the selection pressure, which wiped out those organisms that couldn't now evolve to keep up with the higher oxygen in the atmosphere. Are you understanding what I'm saying? So it became toxic. Originally, when all of this oxygen was getting added into the atmosphere, when Earth was thought to be fully anoxic, and then suddenly there is oxygen in the atmosphere, initially it was very toxic to most of the organisms that was present. Only those that could evolve mechanisms that would protect them from this oxygen completely sort of shutting their metabolism down survived. And that is how you now see that life has predominantly been taken over by metabolism that is driven by oxygen, presence of oxygen. This is not to say that there are not anaerobic organisms. You have a ton of anaerobic organisms to date present in different niches on the earth. People work with them in the lab too. You expose them to even some minimal levels of oxygen. Ke upar, if you expose them, the oxygen will kill them. Deep in the mines, right, several kilometers down in the earth, you actually have people have isolated bacteria from there, and they cannot survive in high levels of oxygen. So this whole saga of some metabolisms being driven by oxygen, some being driven by lack of oxygen, some being driven by methane, still very much happening. And we are still beginning to, we are only beginning to, we sort of, if you look at, I was talking to you about these single-celled organisms occupying all of these different niches that you can think about, right? We have only scraped the tip of the iceberg as far as understanding these single-celled entities. We know very little about them. This is basically called the microbial dark matter, because what we know is just super, super tip of the iceberg kind of metabolism. There is still a lot which we do not understand. But suffice to say, to answer your question, oxygen, more of the metabolism that evolved subsequently went on to be that of survivors that evolved mechanisms to tolerate oxygen, and not only tolerate it, use it as a fuel. Making sense?
Yes, ma'am.
Cool.
Hello, ma'am.
Hi.
I'm audible.
Yeah, you are.
Hello, ma'am. I am Ahitagni Das. Firstly, thank you for the amazing lecture. I have a question, ma'am. Why specifically the hot springs are used for these kind of experiments?
So I went back. Let me just go back to that slide where I showed you the two. Can you see this still on my screen?
Yeah, ma'am.
Yeah. So there are hot springs alone are not the kind of environments that are thought to have supported life. Depending on whom you talk to in the field, they would say that, you know, you're forgetting completely about the hydrothermal vents. If you look at the two of them, both are volcanic systems. One is present at the terrestrial level and one is present on the ocean floor. That is all the differences, right? But what that means is, minus the fact that they're present in two different completely geological niches, the surrounding rock chemistry completely changes. And the fact that they are present way more closer to the, a lot of this activity is directly coming from the magma, and they are way more closer to the magma, right? The kind of gradients that can, temperature and pH gradients that can be supported here, and the kind of temperature and pH gradients that can be supported here are different a little bit, which has implications for the nature of molecules that can form here and here. Having said that, people have very readily shown the formation of, I told you about how a lot of these meteorites struck. There's supposed to have been this period called the late heavy bombardment period, where constantly Earth and Moon was struck by a lot of these meteorites and comets, and they rained a lot of this organic matter relevant to extant biochemistry today, right? Those molecules can also be, like fatty acids, can also be formed under these conditions. Very high temperatures and pressures. If you have carbon monoxide and hydrogen, you can very readily form hydrocarbons. So what that means is you can study these transition processes in both these niches. Personally, for me, this niche is a little less tangible for the following reason. Let's say I somehow figure out to simulate conditions of high pressures and high temperatures and these pH gradients in some contraption in a setup in my lab. One of the things that still I can't get a handle of is, if this scenario allowed for the formation of those protocells that I was talking to you about, as soon as these protocells hit the water, the surrounding ocean water, right, everything gets diluted, complete dilution. Whereas in these terrestrial hot spring-like scenarios, you have all of these surfaces, right? And the interaction with surfaces allows for a lot of other processes. For example, concentrating mechanisms. So if you had this ancestral community of cells, they would have been able to interact with each other much more closely because they are present in a space which is much more constrained and is being sort of held together by these surfaces. So for me, going from the protocellular space to a minimal life space would have been more readily plausible here than in a hydrothermal vent-like scenario. But does that stop me from thinking about questions that might be relevant to ask with these scenarios? Absolutely not. Simulating these conditions in the lab are also completely non-tricky. Imagine the pressures, right? These are the pressures at which your lungs will explode. To be able to set up that kind of a thing in the lab is completely non-trivial. There are a few people in the world that are working working with these, trying to simulate these scenarios. Minus the pressure component, it can't go too much, but suffice to say, the rest of the rock chemistry, the water aqueous chemistry, they try to simulate in the lab, and we are all trying to converge to a potentially, I don't think many colleagues in my field will agree with the statement of mine, but at least I'm very open to eventually coming together to think about, brainstorm about a unified theory of the origins of life, which doesn't say ye yahin pe hua, ye yahin pe hua, right? Because if you have the kind of chemical and thermal fluxes and pH gradients that can be supported in niches that can allow for these things, it could have been plausible in multiple locations. I don't know if I answered that question, but hot springs are not necessarily the only niche. They are just niches that are very tangible for us, and they are also increasingly gaining favor in the field because of a lot of very, very cool things that they support, both at the level of geology, geochemistry, and prebiotic chemistry. I hope
Okay,
that
ma'am.
answers the question.
Ma'am, I am Aditya Mondal. Can I ask you a question?
Yeah, sure. Hi.
Is mammals
Students,
created
please
or
mention.
humans created from mammals?
My dear students, I am interrupting. Please tell about your semester. In which semester do you belong? Okay.
Yeah, go ahead, Arithro.
Last semester, ma'am.
Okay, go ahead.
Once again, please tell your question, Arithro.
Is human created from mammals?
Say that again, Ma. I didn't hear that. Can you repeat that?
Is human created from mammals?
Are humans created from mammals? Is that what you're saying? I'm sorry, I'm not able to hear his question properly. Can somebody else communicate that to me? We are mammals, if that is what you're wanting to understand. We are mammals, and we are thought to have evolved over these billions of years, much, much closer to, mammals were present as far back as 200,000 years ago when dinosaurs roamed the planet. Dinosaurs just reigned the planet because they were just these massive creatures that didn't allow the other smaller creatures to be able to survive and thrive to the extent that they did. However, this famous event, which is thought to have led to the extinction of the dinosaurs, the Cretaceous Ternary mass extinction event, which is thought to have been because of meteoritic impact in a region of Mexico, which led to a lot of volcanic activity subsequently, including in the Deccan Plateau, right on which I'm sitting right now in the Pune region. These two back-to-back episodes is thought to have wiped out the dinosaurs on the planet, which then allowed the smaller mammals. I'm sorry, I don't have that image, but one of our ancestor is thought to look like a mole-like creature. This mole-like creature now started, they used to live in burrows and stuff. They came out and then they started adapting themselves to several other kinds of geological environments and niches and took over the planet. And roughly about less than just literally about in that clock that I showed you, the clock of origins of life, if midnight was when Earth formed and the planets formed, and very close to midnight at around 11 hours, 59 minutes and close to 58 seconds is literally when humans took over. I mean, humans emerged and started to kind of reign the planet, so to speak. So I didn't understand your question fully, but if this is what you're asking, we are mammals and we kind of evolved from much more primitive mammals.
Hi, my name is Mayur Mitra and I'm.
I can't hear you.
I am honest. Is there a network problem, ma'am? Sorry, ma'am.
Ma'am, my name is Mayur Mitra
next
and I'm
question.
semester 3.
Mayur?
Yes, ma'am.
Yeah.
What you told that life about near the hydrothermal vents and hot springs, it's very plausible and understandable and very logical. But my question is, how life? You said that we also have formed amino acids and other organic stuff in asteroids. So but they travel in a very cold region. There's vacuum. There's only water they can get is ice and they're constantly struck by UV radiation, and moreover, when they fall, they receive a tremendous amount of friction through all the layers. So how can we get uh, organic substances from them?
So I will answer your question in the following manner. So 1960s, mid 1960s, or I think it was 1960 itself, this meteorite called the Murchison Meteorite fell in Victoria in Australia. Yeah. So like you correctly say, when it's entering the planet, when it's entering the outermost layers of Earth, there's a ton of friction, and a lot of the outer layers of the cometary interplanetary objects burn off. Yeah, but having said that, the core is still um, pristine and intact. So when you're doing some of these studies with your meteorites, you're not looking at the mineral makeup outside, or you're not looking at the chemistry that is plausible outside, because that would have been completely contaminated by terrestrial chemistry. What you have to do is drill to the core of these meteorites, super deep to the literal heart of the meteorite, extract the organics, and then try to study it. And what they have gone on to show is, you'll be amazed to hear that there are a whole bunch of amino acids with different handedness that have been isolated from not just these Murchison meteorites, but also from other kinds of meteoritic infalls that have happened in the more recent century, last few decades, which actually go on to show that you have several other interesting organics which life has not even sampled. Life on Earth, sorry, let me take it back, that we don't see in extant biochemistry, modern biochemistry. Maybe it was something that it sampled several billions of years ago. And these various selection pressures that I talked to you about would have eventually shaped how evolution happened to what it has happened today. And evolution has not stopped happening, right? It's still, the selection pressures are still working on us, and we are evolving. I do not know how we are going to look several 100,000 years from now, but this is an ongoing process. And the core of the meteorites is what one uses to study the organics of the meteorite, not the outer parts, which are contaminated by terrestrial chemistry. Having said that, the interstellar medium is made up of extremely, I mean, I'll share this link with Swami Kamalasananda, and they can share it with all of you, which talks about the kind of interesting chemistry that is plausible even in everything is not vacuum. There are places where you can potentially have very, very, very interesting chemistry plausible. And I do not know why you say it is extremely cold, but if you look at these star nurseries, right, where you're having the birth of stars happening and the deaths happening, it is associated with massive amounts of energy. So there are spaces where you can have very, very interesting chemistry happening, some of it relevant to what we see on Earth, a lot of it which we still do not understand at all, and we do not also probably, maybe there are signatures around and we do not know how to understand them, but it is still very plausible for that, for those events to happen, because otherwise your meteorites and comets, which are essentially, what are they? They are remnants of planet formation, right? They are remnants of the formation of the solar system. So they are essentially made up of the same kind of materials that the rest of the planets are made up of. So what made Earth unique enough to be able to allow for this transition from chemistry to biology? There's a lot of hypothesis there about us being in this very, very unique position, and our distance from the sun being, there is this famous phrase, right? The Goldilocks zone. We are, I mean, when they look for exoplanets, they are looking for these Goldilocks regions across other sun-like stars. So the idea is that the conditions in these rocky planets, which are in these Goldilocks zones, is just right, that given the interesting chemistry that is possible, both prebiotic and pre-prebiotic, you can have the chance for this to transition to biology. But did all the transition process look like how it happened on Earth? It's anybody's guess. And hence, when people are looking at the whole notion of what is life, how do you look for life outside of Earth, there's a lot of extensive debate that happens about what are the kind of molecules that you look for. We try and look for molecules that we understand the most, and that's a fair and logical place to start at. But that doesn't mean we probably should be only thinking about this. For example, I talked to you about the pre-RNA worlds, where it's thought that these molecules were made up of RNA-like molecules, but the chemistry is completely different. And some of this we understand in the lab when we work from a bottom-up approach. So maybe we can even look for some of these signatures and see whether they are plausible. There's a lot of unanswered questions, and we're trying to answer them sort of slowly but surely. But having said that, it's something that that keeps us excited about our work, and we are beginning to try and I think that is why it's extremely important for us to understand how the process played out on Earth first, so that we can, even now, going back to 4 billion years ago and exactly resimulating the geological environment, then is completely non-trivial. We look at rock records, we look at experimental outcomes of simulations to try and understand what might have been, right? So we have some logical ways of thinking about this, but whether everything panned out in this exact same way and in the sequence of events is anybody's guess, and it's very hard to say. But this chemistry was plausible in the interstellar medium, and a lot of that chemistry did sort of probably initially feed the prebiotic ocean with interesting molecules. But the evolution, subsequent selection pressures and the evolutionary process eventually led to the space that we are in, that is being harbored by living entities that are made up of the molecules that we are made up of. I hope that has slightly answered your question. I'm sure it's up 100 more questions. Yeah.
Ma'am, good evening, ma'am.
Hi.
Thank you for this wonderful lecture. And ma'am, I am Shayan Kumar Ghosh from semester 1, Botany Department. Ma'am, I have a question regarding, you have explained the model for terrestrial origins of life.
Yeah.
And there, ma'am, when after dehydration process, when there is a cluster or the layer forming beneath the pit, or that ma'am, beneath that pit, and after again when rehydration occur, then hydrophilic portions are on the top, then these bulging out portions are because of that hydrophilic part. Ma'am,
Yeah,
actually
so,
they are water loving, so they wanted to go to the water,
yeah,
attached
so
to
they
the
are
water.
the ones that interact with the water. And what happens is, it's just not them interacting with the water, right? So here is a very cartoonish representation of your amphiphilic molecule, right? You have these fluorescent green and blue color heads, right? They're just
Yes, ma'am.
indicating two different kinds of fatty acid and fatty acid derivative containing head groups, like the ones shown here. Yeah. So you have 1, 2, 3, 4, 5, 6, 7, 8, 9. A 10 carbon chain backbone with a carboxylic acid head group. So this is a decanoic acid, and this is decanol, a derivative of this molecule. So this is sort of represented in this cartoonish way here. And what happens is, if you take a whole bunch of these molecules and you put them in water, the water-hating groups want to not interact with water. Driven by that urge to not interact with water, they come close together and they form these kinds of structures. You see that? You see these structures? These are bilayered vesicles. Now, these vesicles essentially...
Like a missile formation, what we use in chemistry for detergent while cleaning, that type of formation occurs, right?
Exactly. Micelles are single layers. Micelles are right here. The thing that you're calling as missile, I'm just using a slightly different way of pronunciation. It's called micelles. Micelles are a single layer where the insides are the hydrophobic entities and the outsides are the water-loving head groups, and they have pushed out water, right? In the case of vesicles, they have an aqueous interior because they're a bilayer. There's a double layer where on either sides you have polar head groups, and when this double layer folds back on itself and forms a vesicle, it can entrap an aqueous interior layer. Just like in our cells outer layer is amphiphilic lipids interacting with some proteins and stuff, but in the inside it is aqueous. 70% of cells is water. And
Yes,
all
ma'am.
of this grand interaction of molecules that you're studying and this crosstalk between different biomolecules is happening in this background of water as a solvent. So this particular thing readily forms as soon as these amphiphilic molecules are put into any space which has water, because of this phenomenon thermodynamic constraint, which
Yes, ma'am.
which forces the water hating groups to come close to each other. And once they
Okay,
come
ma'am.
close to each other, they form these bilayers, and the bilayers, after a certain size, are unstable and they fold back and form vesicles. So they can very readily form.
Okay, ma'am. In this way, it was imagined that terrestrial life was
It is thought that at
originated.
least these protocellular entities came about. And like I said, this inset here actually shows you an experimental result. In our
Okay.
lab, we can readily form these protocell-like entities where we have encapsulated some RNA, some DNA, or some sets of molecules which can interact with each other and form a minimal metabolic network, so on and so forth. And this can be very readily done in the lab.
Okay, ma'am.
Yeah.
Ma'am, I have one more question. Ma'am,
Yes.
ma'am, as you said that at the first slide that before the life was coming, bombardment is there, vigorous bombardment on Earth surface and on the moon surface
Not to
also.
have been there, it is very highly debated. But let's assume it was there.
Yes,
Go ahead.
ma'am. So bombardment occurs due to the meteoroids, and
Right.
it is from the outside space, na?
Right.
So, ma'am, by studying within the core of the meteorite, we are getting some knowledge, some kind of hydrocarbons extracting, and some kind of essential things.
Yeah.
But what is the need, ma'am, of studying those things? It is same on the Earth surface also.
No,
We
no, no,
can.
you're right. You're right. But what we do not see, they are ancient, they are the precursors which led to the formation of Earth. They form the original planetesimals. Yeah.
Okay.
Earth has undergone a lot of geological ripening, if there is a phrase like that. I'm sorry, I'm not a geologist and I'm forgetting the exact term. But even Earth as a planetary system has evolved, and the modern Earth that you see now, the
Yes,
geology is heavily influenced by the biology. Just the oxygenation event, right? What did it do to the Earth's atmosphere? It changed the ratio of the gases present. And those gases now will change the chemistry that is possible, the atmospheric chemistry that is possible. And atmosphere and the surface processes are all extremely tightly interconnected. If you study Geology 101, you will appreciate all of this a lot. So very quickly you will realize that modern Earth has been completely transformed by life. So yes, you
so
can understand a
meteoroids.
few things, but you have to study the ancient original material that formed the Earth to understand what it was 4 billion years ago. Yeah, go ahead. You were saying
Ma'am, that ancient, you say na that ancient material. So ancient, ma'am, that due to this continuous bombardment, only the Earth surface is forming.
No. Solar system formation happened even solar system formation happened is thought to have happened about 4 billion years ago. Yeah, let me just go back to that. So you have formation of Earth around 4.5 billion years ago. This
Yes,
late heavy bombardment is thought to have happened closer to this space. Like I said, please take it with a pinch of salt, because it is being heavily debated by the geology community still, whether late heavy bombardment period was a continuous several hundreds of millions years of bombardment, or did it happen in intermittent phases. Geologists and planetary scientists are still trying to tease this process out, and are trying to come to are still debating about the exact way in which it happened. But all of them do agree that there was much more active period of this bombardment in this phase, which would have reined in a lot of interesting molecules, brought in a lot of interesting molecules to the Earth. And one very important thing that I forgot to mention, all of the water that you see on the planet actually rode in these meteorites, core of these meteorites and these comets. All of the water that you see on the planet today has come from there.
Okay. Thank you, ma'am.
Thank you, madam, for such a long interaction with the students. But a few teachers are still asking for interacting with you, because they have a few questions also. So the meeting is open to some teachers also who can ask you some questions. So with your kind permission, I will ask them to start their questions.
Sure.
Hello, ma'am. Am I
Hi.
audible?
Yeah.
Yes, you are audible.
Yes, ma'am. Ma'am, this is Dr. Krishnendu Ghosh. I am the guest faculty of the Department of Zoology.
Hi.
And I have two quick questions. Number one, since the great oxidation event, what actually limited the level of oxygen to be 21%? This is question number one. And question number two, what kind of differences could be expected in some other icy worlds like the Enceladus or the Europa in its subsurface ocean, if any life could be seen here? Especially the Europa Clipper mission is around the corner.
Yes, it is. Thank you, Krishnendu, for these questions. I do not have the answer to your first question. I do not know the answer yet. There are some hypotheses about what limited this oxygenation, this percentage oxygen to about 21%, 22%. But I really don't feel, I'm completely out of my depth there to answer that question. But I can send you a bunch of resources from this planetary science collaborator of mine, and we'll have an exchange after that over email if that's okay with you. Yeah.
Yeah, definitely, ma'am.
Sure. Coming to the second question, icy worlds are also very fascinating, right? As far as Europa goes, there is a notion that you actually have below all those layers of ice, there are thought to be hot springs that are present. And in this context, studying these hot springs are also very interesting, though, of course, you do not have this icy giant layer above. It is very interesting to still think about what are the possibilities that are available in these hot springs that then allow us to understand some of this hot spring geochemistry and geology and the potential biology that it can host, and then translate that understanding to how we might think about life processes or the transition processes in these areas. One other thing is to also think about what might be plausible in the ice itself, in the ice layer itself. And I told you about how we are very interested in understanding formation of informational molecules. And there was this very pertinent question of why only hot springs, right? Hot springs are not necessarily the only niches. It is just a more easily understandable niche for me, but there are colleagues in the field who have actually looked at ice as a medium to facilitate some of these informational polymer formation. And the cool thing about this ice niches is the following. When we do a lot of this chemistry, and I said it is non-enzymatic polymerization, right? So we are not looking at how enzymes might have stitched the monomers together. We are completely looking at how activated molecules, because of intrinsic energy, came together to form some of these polymers. These activated molecules are very tricky and very temperamental. As soon as you work with high temperatures, like what we think about as Darwin's warm little pond or these hot spring conditions, they fall apart. But if you now invoke ice-like scenarios, ice scenarios where your temperatures are 0 and below, they provide these. You remember I talked about a concentration mechanism where in these hot springs, you go from a 3-dimensional space. During dehydration, you go to a 2-dimensional space where interaction between monomers is more favored. These interlayers of the ice do exactly the same thing. They can, especially if you imagine ice formation with some brine channels, if there's salt around, and salt is around everywhere, right? Your primitive ocean also had a ton of salt. You can actually have, and you've had ice ages form, and there are records of this, geological records of this, and there is thought to be an impending ice age, which is going to happen probably a few thousands of years from now. When this happens, brine channels that are formed in between these ice crystals, actually, if you have solutes relevant to these informational polymer present in the monomeric form, it's been shown that you can readily oligomerize them. And the cool thing about these icy layers is that they are now protective. You don't have these activated molecules falling apart. You can have them survive for their half-lives are much, much longer. And then the argument is maybe you can form much longer molecules in these niches, right? But then one of the questions then is, these ice ages were not very, very protracted. What was more protracted are the kind of environments that I'm talking to you about. Hot, temperatures varying from 60 to 100, 120. These ice ages happened in only small sort of spaces during the history of life of Earth. So yes, it's interesting to understand the chemistry in these scenarios to better understand what might happen when we manage to send one of these Europa flybys and try and understand and bring back samples. I don't know when it's going to be possible and stuff, and when we'll even see samples come back from these other worlds. But understanding chemistries that are plausible in those scenarios will allow us to be more prepared to discern the potential chemistry that is possible in these sort of other worlds that are icy in nature, icy worlds, like you said. I don't know if that answered all of it, but I hope it's given you some flavor of how we think about it.
Definitely, ma'am. Thank you so much.
Thanks. And I'll get your email ID from Kamalasta Nandaji, and then we can be in touch about the other important
Sure, ma'am.
Even this oxygen of 22% didn't happen very readily. There was this dip in oxygen during the ice age periods, and then it came back up again. But what keeps it at this 21-22% for a very protracted period, I really do not know. But there's a lot of geology that kicks in, which then is sort of impinged by the biology too. So it's very complicated, and I don't understand it.
Okay, ma'am. Thank you. Thank you so much.
Thanks.
If there is no more questions, we will move to the next part of the program. I will ask once again. Okay, it is the last question. It will be the last question.
Hello, ma'am.
Okay,
Nice
please
to meet
continue.
you. I am
Hi.
Rajashree Chattopadhyay of first year zoology. Ma'am, although mitochondria and chloroplast reside inside the cell, yet they have a separate genome. Ma'am, what is the possible reason behind it?
So there is this very famous hypothesis or proposal that was put forward by this extremely famous microbiologist called Lynn Margulis. She was Carl Sagan's ex-wife. At some point, the two of them were married. She's a very, very famous microbiologist, and she proposed this endosymbiotic theory, which has now increasingly being proved by experiments and understanding of various aspects of microbial processes. It's kind of it's getting close to like it being accepted as a true possibility, which says that your eukarya formation, right? Your eukaryote formation is thought to have been a product of this endosymbiotic process. You had the archaea engulf a bacteria, which essentially led to the formation of eukarya. I'm sort of oversimplifying this, right? But now this can explain why the genome is so different in the internal compartment. Am I making sense? Rajashree, hi.
Yes.
So this is called the endosymbiotic theory. And this is something that was proposed a couple of decades ago, a couple or more of decades ago. And this is how it's thought that our first ancestors even came about, where one organism from one domain of life engulfed another, and they came together, I don't know, symbiotically, or what happened, merged. It's unclear, but this is why it is thought that the mitochondria and chloroplast have very different genomes.
Thank you, ma'am.
I think that there is no more questions, and we'll be moving to the next part of the session. Thank you, madam, for creating such an environment which has made the interaction so much dynamic, that the topic you have chosen and different participants, including the teachers as well as the students, has continuously interacted during your lecture. It is really a blessing for us that we have got amongst us today. We are blessed to get your thoughts as well as different thought-provoking, enthusiastic topics that will create a research-oriented mindedness among the students. Thank you, madam, for being with us. And now I would request Dr. Kautilya Bhattacharya from the Department of Zoology to please give the vote of thanks. Over to you, sir.
Thank you very much for the kind words.
Thank you, Somir sir, for your statements. First of all, I'd like to thank Professor Rajamani for her beautiful lecture. I had a question, but I was thinking that whether it will be a long, you are talking for a long while, and should I ask you it or not? I was confused. So
you should ask me. Let's see if I can give a very quick non-answer answer. Already I'm giving you answers which throws up more questions, because that's the nature of this process. But yes, please ask your question. Let's see.
Okay, my question is, a very recent study from Nature Geology was stating that by 2100 means 2100 year, they are expecting to have the permafrost of Arctic to be very disturbed, lost, and they are proposing that in that situation, very much along with those radioactive waste that are deposited there in the cold weather situation, number of bacteria which are antibiotic resistant are going to come out, because they have already seen such kind of thing in Siberian permafrost. So my question is, why do you think that those permafrost are harboring those antibiotic resistant varieties?
So just to very quickly answer your question, I think it's a coincidence. See, you could have looked at some deep mine and then isolated a bunch of bacteria, and they would also be probably antibiotic resistance, because they have just not experienced it. You know what I mean? When does something evolve a mechanism that protects it? Only when it is being exposed to it. Now, if the permafrost is all going to vanish and all the methane and stuff that is present in them is all going to come up and is going to completely change the gas ratio in the earth to a point where we will have a lot of difficulty surviving, how do you think you and I will survive? We are humans and we are capable of complex things, so we'll immediately construct a chamber which is very ambient, and this is something about going and colonizing Mars, right? We'll immediately do that, and hence we will survive. So all these microbes that are being discovered as antibiotic resistant, not antibiotic resistant, I'm sorry, is only because they are not antibiotic resistant because they've just never been exposed to that. Even now, they might be resistant to some other molecule about which we do not know. We need to study those colonies of microbes in conjunction, because when does a microbe make an antibiotic? They are constantly, this is called warfare, right?
Yeah, it's kind of alpha selection.
There is constantly other bacteria present in their environment that they're competing with and they're fighting against. They are generating antibiotic as an arsenal to fight them. And along the way, the other bacteria which is now being attacked by this particular arsenal is evolving mechanism to prevent it from killing them. So this is a continuous sort of evolving warfare that keeps happening non-stop.
It's co-evolution kind of a thing.
Exactly. So having said that, I feel like it is because they haven't seen the antibiotics that we know, which is only like not even
small molecules. Yeah,
it's like extremely small number of molecules. We are saying it is full of antibiotic resistant microbes. I think it's just because they haven't been given enough time to encounter these molecules and evolve.
Understood. Anyway, thank you, ma'am, for your nice. So first of all, may I take this opportunity to express our heartfelt gratitude towards Professor SUDHA Rajamani for accepting our invitation and also for this brainstorming session. Thank you, ma'am. Thank you very much. I'd like to take this opportunity also to express our sincere thanks to our principal sir, revered Swami Kamalasthanandaji Maharaj, for conceptualizing and patroning this beautiful webinar. May I put my hands together for revered Swami Vedanuraganandaji Maharaj and Dr. Arunabha Mukherjee sir of our department. Both are our DBT coordinators of Department of Zoology of our college. I extend my sincere thanks to our convener, Professor Somir Sardar sir, and our joint conveners and members of the webinar committee for their continuous support. You you, all the participants, like respected teachers of our college, respected teachers of other institutions, research scholars, and dear students, I thank you all on behalf of our Department of Zoology of Ramakrishna Mission Vivekananda Centenary College for making this webinar a grand success. Last of all, I must acknowledge Department of Biotechnology, Government of India, to award us DBT Star College, under which program this webinar is being organized. And at the end, I thank you all for your patience, for your kind and enthusiastic cooperation. And thank you, ma'am, once again.
You're
Thanks.
most welcome. Thank you for having me. Thank you all for your very kind words, which I don't feel fully deserving of. And
Definitely
yes,
deserving, ma'am.
I don't know, but I hope the Q&A session is never about answering questions, right? It is facilitating a discussion that opens up more questions.
More questions,
and
exactly.
that is exactly what I hope you take away from this, not just this talk, but also from the discussion session. So thank you all very much. Thank you, Swami Kamalasthana ji and Kautilya and Samir ji. I'm hoping I'm getting the names right. Thank you all for your very gracious words, and thank you for hosting me online.
So, ma'am, thank you. So when you come back, you come in Kolkata, please give us a missed call.
Absolutely, totally will do that. I should save your number. I'm actually going to mail and get your number so that I really save it this time.
Yes, yes, yes.
Thank you very much.
Yes, yes.
Thank you. Bye. I'm going to exit if that's okay. Good night.
Yes.
Bye.